Hey everyone, I hope everyone's doing well today. So we are in Lois Pollock's lab, and we use sort of techniques that are rooted in physics and engineering to study the biological molecules called DNA, RNA, and proteins. And so through the course of this workshop, we're gonna hope to convey to you guys the significance of these things. So we've kind of come up with this space / science fiction theme for this. So we kind of like borrow / rip off characters from the Star Trek franchise. So if you're into Star Trek or science fiction in general, hopefully you find this at least marginally enjoyable. Okay, so the title of the workshop is called The Mysterious Message. So we begin here. Oh, also I should mention before we start, ideally everybody should need a pipe cleaner, so at least one pipe cleaner and some beads with letters on them. So hopefully everybody has these things. If you don't, then no worries. We will kind of demo everything, and in terms of like material, it's not overly complicated. So you can still follow along and get something out of it, I think, if you don't have these things. But okay, so we'll just jump right in. So we start here on board the Starship Enterprise, and we're pursuing an unknown vessel. So we start on the bridge of the Starship with our communications officer.
Incoming message, Captain, from a Captain Nucleus aboard an unknown alien vessel. She requests assistance in curing a passenger with a genetic disease.
Very well. Have them send their transmission.
Okay, so this is the transmission that shows up on Lieutenant Uhura's computer console. As you can see, it's just kind of a mess of unintelligible symbols. So we're going to try to make sense of these basically throughout the course of the workshop. So, okay, so we go to the engineering deck now.
This looks like some form of alien genetic code. It reminds me of human DNA. In humans, DNA contains the information your cells need to function. It provides the blueprint from which you're made, from the color of your eyes to the length of your arms. It looks like they use 4 characters for alien DNA. However, in humans, we use 4 letters, A, T, G, and C.
But what can we make out of this message?
Looking into it, Captain. If it's anything like how our cells work, we won't be able to do anything directly to this message. We'll have to further process it.
We better copy it. We don't want to lose the original message.
Excellent idea, Captain. That is just what our cells do. They do not want to lose the DNA message, so they transcribe it into RNA. Although RNA doesn't last as long, it's more functional and can be made into multiple copies. Besides, we'll need a few copies to analyze them here in engineering.
Transcribing message, Captain. One moment. So here I've copied the message into paper and I'm sending it to engineering. You should be able to find this on your worksheet.
Thanks. We'll need all the help we can get translating the message. I've given the copies in your worksheets to our scientist cadets, but they got lost in the mail. So why don't we just use Lieutenant Uhura's original transmission instead? So we can look at it.
To all our scientist cadets, everyone in the audience, please refer to the original, but page 1 of your worksheet, if you have that in front of you. So everybody turn to your worksheet, page 1. To proceed, I think we need to use our knowledge of proteins. Not that kind of protein. Inside our bodies, proteins are molecular workhorses that perform many functions to keep us alive and active. Proteins are translated from RNA transcripts. We might need to do the same for our message transcript.
Excellent idea. In translation, specific orderings in the letters of RNA correspond to different amino acids, the building blocks of proteins. But what do the alien letters correspond to?
I've used all the data available on these aliens and was able to construct the following translation key, which you can also find in your worksheet. If you look on page 3, you'll be able to go through and match them all. I think Suzette will show us more.
Thanks, Lieutenant. With this key, we should be able to translate the message. Scientists, please use the key to translate the message. To keep track of things, when you translate a character, you can insert it into one of the pipe cleaners you have. And that way, I'll dispatch some supervisors to help you.
Okay, I'll turn it over to Suzette real quick to just kind of give a demonstration on how this is supposed to be done.
Hello, everyone. Can you hear me? I hope some of you, if not all of you, have our worksheet and can work with us through translating the message. So if you have your message here, you can use the code that's on page 3 to figure out what this means. So here I have circled the stem squiggle and realized that it's an N. And then the next one is the stem and realize that it should be a, if you find it here, a D. And if you'd like, you can also start writing it on page 4 of your worksheet if you have the worksheet. We've also sent pipe cleaners and beads so you can start constructing the message into your system. So if you have N, you can start putting it in. This is N. Do I have N? And then you can follow it with D and so on. If you don't have this, you can keep going, and actually the message is provided for you. It might be fun to go through it one by one. I don't know if we have time, but the actual message, if you follow this, is here on page 5. So in the end, you should have a full set of pipe cleaners and beads and be able to construct your beads on a string model of a protein system with different letters. So the takeaway message here is you have your signal, your message, you were able to decode it using the code, and you can put it together through your pipe cleaners in this way. So I'm not sure if everybody got the materials, but if not, you should still have the worksheet and translated the message into a following sequence of letters. And if you received the materials, you would have made this bead on a string model with letters on pipe cleaner beads. This is our protein.
All right, yeah, so we'll just jump right back into it.
Great work, people. Do we have the message? What does it say?
Here it is, Captain.
NDXF. Oi, goo. This still doesn't make sense. Engineering?
Well, proteins start as a series of amino acids, like beads on a string, which is later folded into a particular shape that enables function. Perhaps we need to do the same thing and fold our pipe cleaner model to reveal the message. While you were busy decoding the message, I've developed the following folding algorithm. With it, our scientist cadets should be able to reveal the final meaning message. So if you're in the pipe cleaner, so that the first letter N is to your left and the terminal C is to your right, so N and C, in between every X and F, you get to fold to the left with your right hand, and between every Y and G, fold to the right with your right hand. So I think Suzette can show us more how to do it.
So I started with the N on my left side, and I'm folding it with the, I oriented the pipe cleaner so that the first N is on my left and the terminal C is on my right. And in between the X and F, I fold. The Y and G, I fold. The X and F, I fold again. And in the end, it should actually look like this. This is your folded protein. And what you can do is you can actually read the middle column of the letters vertically. And I know not everybody had materials, or you can do this, or not everybody have sorted out their letters, but you can do this in your free time. What's exciting for us is to read what is in the middle, which is our message, D-O-U-D, D-O-U-D-N-A. What does this mean? George?
Thank you, engineering. Please report. Anything tangible?
I've drawn a depiction of what we came up with, Captain. We seem now be able to read the message vertically down the center. This is Dogma.
Dogma. Now that's definitely clearer. Lieutenant Uhura, please search all galactic databases for this word.
Jennifer Doudna, human adult female. In 2020, along with colleague Emmanuelle Charpentier, she won the Nobel Prize in Chemistry, one of the highest awards in science. If you have time later, we definitely recommend Googling her.
Ah, yes. This was a textbook example back at the Academy. Jennifer Doudna and Emmanuelle Charpentier pioneered CRISPR, the molecular scissors that offer the ability to edit human DNA.
Excellent. Tell Captain Nucleus on the other ship that they should try using CRISPR on their patient in order to correct the DNA sequence that is the cause for their genetic disease.
Affirmative, Captain. They have just relayed over their thanks.
Good work, everyone. We were able to decipher the message thanks to our knowledge of molecular biology.
Yes. Molecular biology tells us that DNA, the original code, is transcribed into RNA, the copy, which is finally translated into proteins, the final message. This is known as the central dogma of biology.
This reminds me of the coronavirus pandemic of 2019 to 2021. The exact end date is muddled in the historical records.
Yes, I remember the stories. During the COVID-19 pandemic, humanity created a vaccine that used RNA to make the proteins that enabled immunity. Truly a remarkable feat at the time. Now, on to the next adventure. Navigation, please proceed. Warp Factor 1.
All right, cool. So
Are there any questions?
I have a question to start. So this message that we looked at was obviously pretty short compared to what you would normally be doing. Can you give us an example of like how long these messages normally are in the real world?
So they can be hundreds to thousands of letters in the protein, right? So hundreds to thousands of beads on the string. And it's kind of remarkable that encoded in that message somehow are the instructions for the string to fold up into a protein, and it's the shape of that protein that allows it to do its unique job inside of your body. So really remarkable.
And there's some extra stuff on the worksheet if you want to explore more. So make sure to check that out if you have time.
yeah, the worksheet also dives a little bit deeper into the biological explanations that we didn't get to cover in this skit. And so, yeah, if you do have access to it, then give it a go if you can.
I have a question. So who reads these messages? Messages are stored in DNA, as we learned today, and they get transcribed to RNAs. So they get transferred to RNAs. So we scientists say we did that, but who does it in our bodies?
So inside of our bodies, we have little tiny molecular copy machines. and they're made out of proteins. So these proteins, the molecules that are built up by, by folding these beads on a string, they turn out to make these functional molecular machines. And then those machines have the incredible function. They can actually read the DNA and make the copies. And then other sets of machines, other copiers, if you will, can then read the RNA copy and then translate that message into a string that folds, sometimes on its own into a protein. Sometimes there are helper molecules, right? But you can actually fold and unfold proteins in the lab if you want, right? They're very, some of them are very robust, and all the information they need to fold up, it's written in the sequence. A lot of information in there.
The other thing I'll maybe add, right, is these proteins perform all of the things you need to live. It will help you grow, it'll break down the food that you eat, it'll transport oxygen around your body. So these proteins do all different jobs, and that's just based on what the shape is. So that's why these messages are really, really important.
What type of research do you guys do with RNA and DNA and proteins right now outside of the general overview that we saw?
So we try to understand how these molecules work. So they're little tiny molecular machines, if you think about it. So we like to understand what parts move and how they fit together and how one piece joins with another piece. And that new molecule that's made from two smaller molecules that comes together can have a brand new biological function. So we are very interested in how these machines work. And we're also interested in why they don't work. So if there's a problem and you have kind of a typo in your DNA code or a letter that's not the letter that most everybody else has, you might make proteins that fold in a different way. And in some cases, that can be responsible for some types of sicknesses. And so by understanding the cause of the sickness, we really hope to be able to make better ways to cure it, new types of cures. So our goal is to understand how these molecules work so that we can understand what keeps us healthy, but also to gain insight into things that make us sick so we can prevent people from getting sick. That's the end goal. The question is, how does the mRNA COVID vaccine work? That's a very important and very current question. So the way the vaccine works is that when you get a shot, you are adding a message into your body, and that message codes that turns into a protein. Your cells make a protein that's on the outside of the coronavirus. It's like if you have a Christmas tree with ornaments on it, right? If you think about a virus as being a big assembly that's decorated with things on the outside, and we've all seen these pictures of the spike protein on the outside. What the vaccine does is it teaches your body how to make one of the ornaments. You're not making the whole virus, you're just making one of the ornaments. And then when your body sees that ornament, it realizes that that's not something that your body normally makes. It's not supposed to be there. And your body rallies its defenses to take the ornament away and put it in a trash can, just get rid of it. And so by training your body to recognize just the ornament, you get strong and you get ready. So if unfortunately you do get exposed to the real virus, when the virus comes in and it's decorated with some of these ornaments, your body knows that it's not supposed to be there. And it already has the tools to take it and put it in the trash can so that it doesn't infect you. So the vaccine just gives you the information for making a teeny part, an ornament that's on the outside of the virus, not the virus itself.
So the next question, I think, is how come so many people have had hyperreactions? So like Lois was saying, your body learns how to do its defenses, but sometimes it does its defenses too much. So then your body just gets a little bit run down, and it just gets, you feel a little sick, you feel a little tired, but it's not because you're actually sick with the virus. It's just because your defenses are working on overdrive. So you just get a little worn down and you might need a day or two to recover, but then you're totally fine. That's what's happening there.
That's kind of like the beauty of vaccines. You need it's what makes it work is that your immune system has to fight something. So if it fights it too much, then you're definitely becoming immune, but it does give you a little bit tired. But it's unlike a virus, is very temporary, right? It's like you put that RNA that is going to last 1 or 2 days, and then afterwards your body doesn't have that. So it's very brief, but it just gets you a little sore arm and a little bit maybe like tired.
So the Johnson & Johnson vaccine is very different from the mRNA vaccines that we've been talking about. The way the Johnson & Johnson vaccine is closer to exposing your body to the whole virus. It's not exactly the coronavirus, but it's a different virus, right? So it's a very different process to learn the immunity. And honestly, I don't think scientists know the exact reason why there are blood clots, but it's something that they're looking for and something we're being very careful about. But luckily, the Pfizer and the Moderna and these mRNA vaccines that you guys have probably heard a lot about don't have the same mechanism as the Johnson & Johnson vaccine, and they have not been linked to any blood clots.
And so that's why they've stopped, because everybody's being super, super cautious. So until there's a little bit more information, they've paused the delivery of the J&J vaccine just to understand what the risks really are.